أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم ويل للمطففين إذا اكتالوا على الناس يستوفون وإذا قالوا أو وزنهم يكسرون ألا يظن أولئك أنهم مبعوثون ليوم عظيم يوم يقوم الناس لرب العالمين كلا إن كتاب الفجار لفي سجين وما أدراك ما سجين صدق الله العلي العظيم. So first year class, how enzymes increase rate of reaction we are studying, and what are the factors who increase the rate of reaction, and how we can increase rate of reaction, rate of metabolism, rate of anabolism, rate of catabolism in the living thing. And what are the factors which can stop this reaction, and what is the adverse effect when enzyme is not properly working, is not changing the reactant into product. One disease already I have told you, Tay-Sachs disease, SACH disease, which is caused due to accumulation of the lipid inside the cell. The lipid concentration will increase inside the body. What will happen? The lipid catabolism will not take place. The lipid catabolism will not take place. The lipid concentration will increase in the cell. Now you will get idea that these organic molecules, lipid, protein, fats, lipid is also called the fat, the carbohydrate, they are very useful. They are very useful for the human body. But when they are in excess amount, they are also harmful. Like lipid, when lipid is above normal level, enzymes are required to convert lipid into fatty acid and glycerol. This lipid extra concentration will damage the nerve impulse, of nervous system will be affected, and disease is called the Tay-Sachs. So factors which affect the enzymes, there are many factors. Sometime even the enzymes are available. This is interesting. Sometimes enzymes are available and even the ES complex are not being formed. Even the ES complex is not formed. I will tell you step by step, but here if you will see, first of all, a normal thing I will tell you, because these are normal things which I am just telling you. Okay ji, you are going to increase substrate concentration. This is very normal thing, because this is not any difficult and conceptual thing. When you will increase the substrate concentration, mean more substrate is able to combine with active site. Clear? More active sites are available. The more active sites are available mean more ES complex will be formed. More ES complex will be formed mean more products will be formed. It means the rate of chemical reaction will be the fast. It is clear? Very easy. Another rate. More enzymes are present, more active site is available. The more active sites are available, more active sites will be able to react with the substrate. More active sites are able to react with the substrate, more ES complex will be formed. More ES complex will be formed, they will be able to convert into the product. So dear students, if you see the substrate concentration in this graph, along x-axis is the substrate concentration, and along y-axis if you see the reaction rate. And what is happening here, the substrate concentration along x-axis, along y-axis is the reaction rate. Enzyme concentration we ignore for some time, and we just see that when you are increasing the substrate concentration right from here to there, just see the colors, black color lines are telling the substrate concentration was less, the reaction rate was less. How you can see reaction rate? Reaction rate you can see from here. This is the reaction rate. So reaction rate, substrate concentration less, reaction rate less, substrate concentration more, reaction rate more. This is very good. This is a straightforward, this is a directly proportional relationship. No worry, no tilted information, straightforward information. You agree, I agree, no issue. We'll go back to next part of the graph. Substrate concentration you are going to increase once again. what will happen? The rate of reaction will increase, no issue, this is well. Substrate concentration increased, rate of reaction increased, okay, up until now we are satisfied. We'll go back to next part of the graph. Here is the problem. Now I am touching the problematic area. This is the area in which we are getting a little confused, because you are increasing. The first line is telling the substrate concentration is less, next line is telling substrate concentration is more. Sir, how the substrate concentrations are increasing? I tell you, this is x-axis. When x-axis I will move towards the right side, it means I am increasing the substrate concentration. You can see here is written substrate concentration. So when I will move towards the right side, it means substrate concentrations are being increased. Substrate concentration are being increased. Substrate concentration will increase. So what will happen? You are moving towards the right side, substrate concentration increasing. Of course, there is no any problem. There is no any, until now there is no any problem. But why this graph did not move up? This is a question. This graph was moving up. This graph was moving up, and this graph must move up. Reason is that because you know that when the substrate concentration will increase, the here along y-axis is the rate of reaction. The rate of reaction less, rate of reaction more, rate of reaction less, rate of reaction more. So whenever the graph will move up, it means rate of reaction is increasing. So when you are increasing the rate of reaction, when you are increasing the substrate concentration, this is the substrate concentration you are increasing, the rate of reaction should increase. Rate of reaction should increase. Rate of reaction should increase. But here in both graph, what is happening in both graph? In both graphs, rate of reaction is not increased. So what happens to rate of reaction? What happens to rate of reaction? Rate of reaction here is constant. Look, this line is exactly along the, how I will tell you, just hold on. I minimize the graph and I will show this line. Now I will indicate, and you will see from y-axis this line is telling the rate of reaction is constant. Here the rate of reaction increasing, moving up, but here constant. Here moving up, but here constant. Here is the problem, here is the problem. Here is no problem, it's okay. So what is the issue going on? Here substrate concentration increase, reaction rate increase. Concept is very simple, because substrate will react with the active site to form the ES complex. ES, whenever I will say, that means enzyme substrate complex. Enzyme substrate complex is called ES complex. The ES complex will be formed. That will change into the product, and that is very easy. More substrate you will give to the living thing, to the body, the more substrates will attach with active site. But when here in this line, when substrate concentration increasing, rate of reaction is not increasing. I am pinpointing this point again and again, because this topic resides with this point. Only key point of this topic is that which I am discussing. Now rate of reaction is not increasing. The rate of reaction remains constant. When in this line, after this line, the rate of reaction remain constant. Cold water. Now I will tell you one thing. What happened here? After that, you are alert now, because I am telling you the reason. Now I am telling you the reason why this happened. You must be alert. You must be attentive mentally. You must be right man. If you are listening this clip in the evening, just close and listen in the morning. Or if you are doing any activity, don't listen this, because this is the point you have to learn. Okay. After this, substrate concentration was increasing, no doubt about this, because substrate concentration is along x-axis. But we did not know. We know substrate concentration is required. Is there any other thing also required to form the ES complex? Of course, we require another thing also to form the ES complex. Which thing we require? We require active site. So two things are required. One is active site, one is substrate concentration. So we were just closing the eyes that I am increasing substrate concentration and ES complex will be formed, and I was very happy. I was very happy that I am increasing substrate concentration and active sites will attach more with the substrate and more enzyme substrate complex will be formed, the more products will be formed, the rate of reaction will be more. But I did not give attention to active site. Now another problem, sir, if active site issue was there, if active sites also required, so why here I was increasing substrate, the graph was moving up and rate of reaction was increasing, and here it is not increasing, here it is increasing, here it is not increasing. What is the problem here? There is no problem here, here is the problem. I tell you, look here, if you have 50 active sites, if you have I will give you example, you have 50 active sites, clear, and you have 20 substrate concentration, you have 20 substrate. I am giving you concept, please be alert, because you can revise this clip if you have any problem, you know, because I will speak slowly. This concept will train you that why the rate of reaction was constant. 20 mean 20 substrate concentration. You will increase substrate concentration from 20 to 22. Rate of reaction will increase because active sites are available. You will increase up to 30. Rate of reaction will increase because active sites are available, because active sites are 50. You will increase up to 50. Rate of reaction will increase because active sites were available. When you will increase more than 50, mean that was the 51 substrate and active site is a 50. So rate of reaction will remain decrease? No, it will not decrease. Increase? It will not decrease. It will remain 50-50 because out of 51 substrate, 50 will combine with 50 to form the ES complex. So then after 50, if I will increase even 1000 substrate molecules, only 50 active sites will be available to form the ES complex with only 50 substrate. Reason? Because there are active sites 50. Active site is a limiting reactant now. Active sites numbers are less. So it means in this graph, now you got the idea. Now in this graph, here when you was increasing substrate concentration in the right direction, the rate of reaction was increasing. Rate of reaction was increasing in both graphs. But when active sites, all active sites were occupied by substrate, when all active sites were occupied by the substrate, rate of reaction remained constant. You got the right idea. It means that only substrate concentration is not required to increase the rate of reaction. Which things are required? Active sites are also required. Another point came out of this discussion, that active sites, this was the reason that enzymes are catalysts. So it means that active sites after catalyzing one reaction is able to catalyze another reaction. If active sites would not available for another reaction, our body will not be able to do multiple and many reactions. So you can see here active sites are not available, and active sites are not available, the enzyme reaction will remain constant. Now another point, you was listening my lectures in the last session. I was talking to you that medicines are also related with enzyme, the diseases also related. Diseases I told because lot of diseases are caused due to abnormality in the enzymes. When you give drug medicine, increase the rate of reaction or decrease the rate of reaction in different cases, because there are some nervous diseases in which you have to decrease the rate of reaction of enzyme because human become hyperactive, so you have to decrease. When in psychological patient, in some anxiety patient, in hallucination, they give such medicines. So you have to increase substrate concentration from outer source in the form of the drug, automatically rate of reaction will increase. So enzyme knowledge is not helping us to identify the disease, also helping to form the medicines. So I hope today's lecture will work, and upcoming session I will tell you further points how you can increase the rate of reaction. I think these are very precious informations because they are related with our body. Because if you want to become more active, if you want to get good marks, if you want to become good player, if you want to become a good researcher, investigator, you want to become good doctor, your enzymes must be working properly. Thank you.